And like the other speakers, I want to thank Ann and everyone at the Duke Lemur Center for the invitation to come and participate today. This has been an absolutely incredible symposium, and I congratulate all of you on that. And just as importantly, I want to congratulate everybody at the Duke Lemur Center for 50 years of truly outstanding work. The Duke Lemur Center is a jewel in the community of research biologists, and I think that the quality and the breadth of talks in the last, yesterday and what we're going to hear today illustrates both the ability of the Duke Lemur Center to support a variety of different researchers and the quality of the research that you produce. So I want to congratulate everybody at the Duke Lemur Center for 50 years of outstanding performance and much, much more in the future. Thank you. So what I'd like to do this morning is tell you about work we've been doing on sequencing the genome of the mouse lemur, Microcebus murinus. Now, the mouse lemur genome is about the same size as the human genome, so it's about 3 billion base pairs of DNA, which makes it a challenge to sequence the entire thing, assemble the entire thing, and then figure out what it means. But we've developed a bunch of different techniques that I'm going to tell you about briefly that have allowed us to do this. We've made, I think, some pretty interesting progress. First thing I need to do is tell you who's actually doing the work. I'm very pleased to be the spokesman for this group here today, but what you need to understand is that every time through the talk that I say we did something or we found something, what I really mean is that these folks up here were central to the entire process, and there's actually more people at the Duke Lemur Center and more people at the Baylor Genome Center, but I didn't have room for all the names. The 3 people I want to particularly recognize are Peter Larson, Ryan Campbell, and Alan Harris, who did the bulk of the analyses that I'm going to talk about today. So it's really me functioning as their spokesman. The goals of this collaboration are really to produce an accurate and complete as possible, reference genome sequence for Microcebus murinus, and then we want to annotate that genome sequence to identify all the genes and the protein-coding genes and non-coding genes. And then, of course, the ultimate goal is to make that genome sequence available to the research community so that we ourselves and everyone else can make use of it to expand the research that we're doing on mouse lemurs and expand our ability to conserve mouse lemurs. So to begin, I need to tell you a little bit about how we sequence and assemble a genome. And this is meant to represent the chromosomes of a kind of generic primate species. And of course, we would love to be able to just start from one end of a chromosome and sequence right through to the end of the chromosome, but we don't have the technology to do that. So what we do is we break the genome up into pieces. Sometimes we break it up into fairly large pieces, sometimes into much smaller pieces. But the point is, you take these random pieces of DNA and you sequence in from the 2 ends. What that allows you to do is generate information on one end and the other end, and then you do that again, and you do it again and again and again, millions of times. And ultimately, what you produce is enough reads that overlap each other so that you can generate a complete continuous sequence from all these overlapping reads. This is what we call a contig, for continuous sequence. And that's a term you're going to hear a lot in the next few minutes. So this illustrates, again, we do these small reads. We try to find the overlaps between them and develop contigs. These contigs, we want them to be obviously as long as possible, but you really can't sequence and the entire sequence together as one contig. What we do is use the fact that we've sequenced a random piece of DNA in both ends, and we can know the distance and the orientation of those 2 ends, to connect different contigs into what we call scaffolds. So the scaffolds are basically a cluster of contigs where we know the order and orientation of the contigs. Ultimately, what we want to do is produce a continuous sequence, but our building blocks are the contigs and then the scaffolds. So I have to introduce one statistic here so we can talk about results, because our goal is to produce the longest possible contigs with the fewest gaps across the genome. The way we evaluate how well we're doing in that is the number we call contig N50. And that's basically, once you've sequenced all these contigs and assembled all the contigs, you line them all up and count how much sequence you have in total. Then you order the contigs from the largest down to the smallest. and you ask, how far down the list, to what size contig do I have to go to get half of the sequence that I've generated down above that level? So half of the sequence is in contigs larger than this. That's the contig N50. So what does this mean in terms of real genomes? So the first non-human primate genome sequence was the chimpanzee genome, where the contig N50 was about 15,700 bases. So half of the genome was in pieces 15,700 bases or bigger, and half of the genome smaller. The gorilla genome wasn't quite as good. When we published the rhesus genome in 2007, it was about 25,700 bases for the contig N50, and recently the marmoset was about 29 kb. Now you can go back and you can add more sequence and you can improve these genomes. So the chimpanzee genome has been upgraded from 15,700 bases of the contigs up to about 50 kb, and the rhesus up to about 54. This is important because the average gene in the genome is about 20 to 30 kb, about 20 to 30,000 base pairs. So if your genome sequence is broken into pieces that are 15 or 12 or 10,000 bases, most of your genes are broken up into various different pieces, and you can't really understand the whole sequence. You want to get your contig N50 as large as possible. So now some history about the mouse lemur genome. The first effort to sequence the mouse lemur genome was published in 2011, a large consortium that sequenced 29 different mammals, but they did very low coverage sequence, and I won't go into the details of it. But the contig N50 initially was 3.5 kb, or 3,000 bases. So really lots of sequence, but all in tiny little pieces. What we did then was revisit this genome, and a couple of years ago we started working with Anne and her team down here to resequence the genome with the Illumina technology, and we were generating 100 base pair reads at the end of these sequences. What that produced was much more of this kind of information, so we could build longer contigs. But then Adam English at the Baylor Genome Center developed a new technique for actually improving the scaffolding as well. And that is what he calls PB Jelly software. And this makes use of a different kind of technology. This is Pacific Biosciences long read technology. And the Pacific Biosciences does sequencing a completely different way than Illumina, but I don't want to go into all the details. I can talk with anybody about it later on. But basically, back in 2011, Pacific Biosciences technology could produce a continuous read of about 2,000 bases long. Not the end sequencing, but a continuous read of 2,000 bases. But they've improved their technology so that now we can generate reads that are more than 20,000 bases long, a single read. Now you might ask, well, why don't you do all your sequencing this way? And the reason is it's very expensive and it's actually very error prone. So there are reasons why we don't do whole genomes this way. But basically what this allowed us to do is use the very long PacBio reads to span across gaps in the scaffolds to close many of these gaps. The result was that we did 120x coverage, meaning we resequenced the genome equivalent of 120 times to get all those small reads, built the contigs that way, then improved the scaffolding with the PacBio reads, the long reads, and that increased the contig N50 from 3,500 up to 182,000 bases. I hope you can appreciate that that now tremendously improves the quality of the sequence and our ability to find the genes. But we weren't done there, and this is where Peter and Ryan and Alan really come into the picture in a big way. What I've illustrated here is the Illumina short read sequencing in addition to the PacBio sequencing that produced our second generation mouse lemur genome. What Peter and Ryan and Alan did was work out this part of our procedure here, and I'll just briefly go into this because I don't want to go into all the technical details, but basically what we realized is that we needed better ways of doing scaffolding, better ways of clustering the contigs so that we could then use the PacBio long reads to fill in the gaps. So what they did was make use of a technology that we didn't develop, but we used in this project called Hi-C for scaffolding. And basically what I need to do is remind you that DNA in your cells is not just naked DNA, but it's actually wrapped in proteins. And those proteins wrap around the DNA, and that's what makes chromosomes. So people have worked out a method of taking the proteins that are wrapped around the DNA and cross-linking those proteins. And what that does is allow you to take, this is meant to be a loop of DNA, a long loop of DNA, where the proteins that are bound to the DNA are cross-linked. You can then cut the DNA, stitch it back together, and you know that these two pieces of DNA were close together along the chromosome. This is meant to be a complete chromosome. And what happens when you do this is that the pieces that you connect using this method are generally very close together. You connect the close pieces and you don't connect pieces far away. This allows you to build up the scaffolding. However, the problem is that this cross-linking sometimes, most of the time, occurs within a single chromosome. Sometimes it occurs between chromosomes. So the scaffolding here can make errors. What Peter and Ryan and Alan did was work out a way to use optical mapping, which is another technology, to check and correct the scaffolding that they did with Hi-C. And what optical mapping does, very briefly, is that the folks at BioNano Genomics worked out a way to draw out very long, thin, very long molecules, single molecules of DNA through these wells in a flow cell here. These blue lines are meant to represent individual DNA molecules. Then they nick the DNA, break it a little bit, and label at particular places along the DNA sequence. And you're labeling at a place where the base pair sequence has a particular sequence. And what this does is essentially barcode these different DNA molecules. And when you do this in millions of different molecules, you can read the barcodes and line them all up so that you get a continuous sequence based on all these little spots that you've identified. That allows us to check the contigs and the scaffolds and make sure that we're correct. And I'm just going to skip that. So basically what the guys did was take our initial assembly and expand it and improve it using their new method. And that produced this result. So we've now gone, in just a couple of years, from a contig N50 of 3,000 bases up to 234,000 bases, or a 66-fold increase. And of course, the longest scaffolds have increased tremendously as well. That makes our genome much more valuable for all the kinds of interesting biological questions that we want to ask downstream. This is just another way of representing that improvement, where this is the length of the individual contigs, and this is the ordering of those, the scaffolds, the length of the scaffolds, and we're ordering scaffolds from the longest scaffold down to the shortest. And what you can see is that the longest scaffolds in our new 3.0 assembly are dramatically longer than that first version, and even dramatically longer than the second version. So we've tremendously improved our quality of our genome. And what we're now up to is something close to whole chromosome level scaffolds. That's our goal, is to produce whole chromosome level sequences with no gaps. We're not there yet, but what this diagram represents is all the human chromosomes represented as different colors along this side of the chart. And these are all the mouse lemur, we'll call them Hi-C or lachesis groups. These are the large scaffolds that were assembled and then checked and corrected by Peter and Ryan and Alan. And what you can see is that in many cases, we've got essentially a one-to-one mapping across the entire human chromosome here to an entire scaffolding group here. So another case up here in Peach, where we have a very large mouse lemur sequence that maps to a very large segment of a human chromosome. And you can see across. Now, there are a few places where things are broken up. We still have some more work to do. But this is, I hope you can appreciate, a tremendous improvement in our ability to read the sequence and analyze the sequence. So the question becomes, what did we find when we tried to analyze that sequence? The best way to find the genes in this random DNA sequence across the mouse lemur genome is to sequence RNA, which is produced from the DNA and is going to go on to code protein. So Anne and her team sent us tissue samples from multiple different tissues. We used these 9 different tissue samples, and at Baylor we did deep RNA sequencing to identify what the genes are. And then we mapped the reads from those RNA sequencing to the DNA sequence so that we can figure out where on the chromosomes the genes are actually located. And we then transmit that data to the folks at NCBI, who actually are the experts in doing that kind of annotation. And they found in the mouse lemur 2.0 genome, so not the latest one that Peter and Ryan and Alan produced, but the previous one, 20,000 protein coding genes and 6,700 non-coding genes. So this is really a very strong annotation for this genome. And when we looked at the mouse lemur 3.0 version of this, what we found is that when we tested 15,000 genes, 96% of them, of the genes that we looked for in the 3.0 version, were actually present in that genome at a length of 95 to 100%. So not only do we think we have a high quality sequence, but we have a very complete sequence of the mouse lemur genome. So this lets us really move on to the next phase, which is the really interesting phase, which is doing something interesting with this genome in terms of understanding the biology of mouse lemurs. And that's the stage that we're at now. Anne and Kim Worley, my colleague at the Baylor Genome Center, and I have organized the International Mouse Lemur Genome Analysis Consortium. And this is right now a group of more than 50 investigators from 9 different countries. And we've brought together, very intentionally, experts in comparative genomics who are experts in reading this DNA sequence and understanding how it functions, brought those people together with experts in the biology of mouse lemurs, because ultimately the purpose of this genome sequence is to help us understand the biology of mouse lemurs. And we, over the next few months or a year, this group is going to be working together to fully analyze and interpret the mouse lemur genome sequence that we've produced. This is not a closed group. We are open to anyone who wants to participate. And I know some of you here in the room are already members of the consortium. Any of you that want to participate, we welcome your participation, because the more eyes and the more hands working on these data, the better job we're going to do in this analysis. Specific topics, as you could well imagine, that we're interested in working on are things like the genetics and the physiology that underlie torpor, or aging in mouse lemurs, the neurobiology. David Weisrock talked really interesting information about speciation and genetic differentiation. We want to look at that at the level of whole genomes. We want to look at population genetics and conservation genetics. And one of the really interesting things that we think we can do is, now that we have the whole genome sequence in all the genes in the mouse lemur genome, we can now investigate whether there are genetic models of human diseases where mouse lemurs can make a unique contribution to our understanding of biomedical research, as well as the fundamental basic biology of mouse lemurs. And then something that I'm interested in, I know Anne is interested in, is the ancestral primate genome content. We now have a very good sequence for humans, for great apes, for old world monkeys, new world monkeys. Now that we have an excellent strepsirrhine sequence, we can ask, what kinds of genomic innovations were there that actually led to the origin of non-human primates? And I think that's going to be a very exciting topic for the consortium. So the consortium is just beginning. I don't have any really results from the work of the consortium yet to share with you, but I want to share with you 2 different aspects of the analysis that we've started in my lab. And these are very preliminary data. The first batch is a little bit old, a few months old, but the second set of data are completely new. Literally, I was getting data on the plane as I was flying down here. So some of it's kind of preliminary, but I think you'll find it interesting. At least I hope you will. So the first thing we want to look at is genetic variation within mouse lemurs. We resequenced, so did a complete genome sequence of 5 additional animals from the Duke Lemur Center, 5 from the Montpellier Center in France, and 5 that were sent to us by Mark Krasnow from Stanford, but actually came from the Brunois colony. And the details of this are, we sequenced with Illumina, we mapped the reads with BWA, and we did SNP calling with GATK. Anybody who wants more information about this, I can talk to you later. The important point is that when we looked at the data, we found 37 million places in the mouse lemur genome where these 12 individuals were different from the reference sequence. On average, each of those animals is different from the reference sequence at more than 6 million places. Now, that's just a number, but I'll tell you that in human genetics, it's very well established that for the human reference sequence, each of us are different from the human reference at about, on average, 3 million places. So there's more genetic difference between these mouse lemurs and the reference sequence than there is across different humans. And here's another way of looking at that information. This is a very important paper published on great ape genomics a couple of years ago. And basically, this is a graph. You probably can't read these names here, but this is a graph of heterozygosity, the amount of variation in a population. And this is humans from people from Africa and people from Europe and Asia. This is the level of genetic variation in humans. This is bonobos, Western chimpanzees. A little higher level of genetic variation are the other subspecies of chimps, and the highest among the great apes is the Sumatran orangutans, and we could talk about why that's true. But in my lab, we've sequenced about 200 rhesus monkeys, and what we found is that Indian origin rhesus monkeys have about this level of heterozygosity, and Chinese origin rhesus have about this level, so higher than any of these apes. Now we look at the mouse lemurs, and we see that the mouse lemurs are twice as much variation as humans and bonobos and other chimps, and right in line with the highest of the great apes. So this is really interesting because it tells us that there's a tremendous amount of information to be gained from resequencing more mouse lemurs, learning more about what kinds of polymorphisms, what kinds of functionally important differences there are. Now, the last thing I want to talk about is the brand new data, and this is going to be a little bit tricky to explain, but I want you to work through it with me. This is a way of using whole genome sequences to look backwards in time. Now, you can appreciate that large populations, whether it's a large population of macaques or mouse lemurs or humans, a large population can accumulate lots of genetic variation and sustain it over time. A small number of individuals in a small population doesn't accumulate as much genetic variation. This is a very well-established principle in population genetics. So we can estimate the population size of a natural population without counting the number of individuals by looking at the amount of genetic variation that's segregating in that population. Two population geneticists, Richard Durbin and Hung Lee, worked out a method for using whole genome sequence information just from a single individual, from one person or one animal, to work back in time to see how much genetic variation was in the population at past intervals. And basically what they're doing is taking the two different chromosomes you get from your father and your mother and comparing how much difference there is between those two chromosomes. But by looking at the pattern of those differences and how they're distributed in different blocks, Lee and Durbin worked out that you can actually look backwards in time. So this is an example of an analysis we've done using this method in baboons, where we've looked at 6 different species of baboons, whole genome sequences. And the way to read this chart is that this is the population size. This is the estimated population size. And this is the very recent time, and this is looking backwards in time. So this is about 1 million years ago. This is about 100,000 years ago. And this is the population size. But what I have to tell you is it's not the census number of individuals, but it's the effective population size. The effective population size makes some assumptions to make the mathematics easy, but it's going to be highly correlated with the census population size. So we're not actually counting the number of individuals that were present, but we're estimating in a very accurate and reasonable way that I could go into more detail if people want. But basically what you can see is that all the baboon species share population size down to about 2 million years. Then it increases in size, and then they begin to split. And we think this is when the different species of baboons started to differentiate, where the northern species, here, these 3 lines, lose their population size, and we think this is when the baboons that evolved in southern Africa dispersed north. So we can read backwards in time the history of population size increases and population size decreases. Now, we've done this initially as a first pass preliminary data for the mouse lemurs. This is one of the sequences from Duke, not the reference animal, but a different animal. And what you can see is that here we have the population size measured in tens of thousands. So this is 100,000, 200,000, 300,000. And this is measuring time. So 100,000 years, 1 million years, 2 million years, 3 million years. And what you can see is that around 2 million years ago, the population size was around 100,000, and then it increased until about 800,000 years ago to a peak, and then it declined. Now, you need to ignore this stuff over here. This method is not accurate in the most recent time periods, so when you see this, we don't believe this kind of result. But what's interesting is that when you look at chimpanzees, chimpanzee history shows that the effective size has never increased above about 50,000, and the human effective size has never increased above about 25 or 30,000. So mouse lemurs have higher effective population size than humans or chimps. We then sequenced the Montpellier sample and the Stanford sample, and what you find, reassuringly, is that those two lines overlap until the very recent time, suggesting that our method is repeatable and reliable. And then we add back the Duke sample, and you can see that all three of them really line up. So we think we're beginning to really understand the demographic history of mouse lemur murinus using this technique. But Anne and her team also sent us samples from Grisea rufus, from Mittermeieri, and Taberatra, and a sort of currently undescribed species from Mononara Nord, as well as an outgroup from Mirza. We've now done whole genome sequencing for all of these animals, and what we find is, I think, quite interesting. What you need to do is read through the red and the green are the gray mouse lemur, the murinus. That's coming here through the graph. So 1 million years ago, they were about 150,000 in their effective size, and then it declined and it goes back up. The Grisea rufus are exactly the same until about 1 million, maybe 900,000 years ago. And then the Grisea rufus expand to be about twice as large a population size as the murinus. On the other hand, the Taberatra, they seem to decline and then recover. Now, there are people in this room who are much more expert in microcebus biogeography and evolution than I am. So I will defer to all of you. This is why we have a consortium to analyze this genome, because we need you to help us interpret these data. But my sort of initial understanding is that murinus right now has a pretty broad distribution. Grisea rufus has a fairly small distribution geographically. But of course, this implies that 700,000, maybe 800,000 years ago, Grisea rufus probably had a much broader geographic distribution, and that over time it has actually declined where murinus has actually increased. So I hope you can sort of begin to appreciate that this genomic information is going to give us a lot of new insights into the biology of mouse lemurs that we might not be able to achieve in any other way. The next thing I want to talk about is kind of more of the biomedical implications. And this is our interest in understanding genetic variation within murinus as a way of developing new models for human physiology and human disease. Ultimately, what we want to do is compare the mouse lemur gene sequences to all the other primates so we can understand what's different about mouse lemurs from other strepsirrhines as well as other anthropoids. But we also want to understand phenotypic consequences of genetic variation within murinus itself. And I'll just show you some initial results here. Once again, this is brand new data and it's preliminary. We're going to do some more banging on this data to make sure we believe it. But Alan Harris in my lab has looked at the sequences of 18,000 genes using those 12 whole genome sequences, 5 from here, 5 from Montpellier, 2 from Stanford. And what Alan finds is that there's 30,000 mutations, 30,000 polymorphisms, which we call missense mutations. These are variants that change the protein sequence of the gene. So the predicted protein sequence would be different. And that's 30,000 different examples just in 12 animals. Each one of those becomes a potential model for understanding how that gene can influence individual variation in physiology, in anatomy, in growth and development. And the other one that I want to point out is the stop gains. 438 times we find a mutation in the 12 animals that seems to stop the translation of a protein. Essentially, it's a natural knockout mutation in that gene. And we think that these can be extraordinarily important for understanding how genes function in mouse lemurs. So this is the kind of thing that we want to follow up through the consortium and through what I hope will be a very long and productive continuing collaboration with the Duke Lemur Center. So with that, I want to point out that our future plans for the consortium, as well as beyond the work of the consortium, are to identify more genetic variation within Uranus itself, but also within all those other species, and apply that to evolutionary genetic analyses at various different levels. We want to look at whole genome analyses of species diversity and species phylogeny, and work with Dave Weisrock and others to develop a better understanding of the phylogenetic history of this genus. And then we want to look at functional studies that connect genotype and phenotype, both in terms of the adaptations of mouse lemurs in torpor and in aging and various aspects, but also their potential biomedical applications. And then finally, I'm very interested in reconstructing the ancestral primate genome content. So with that, I want to remind you that it was Peter and Ryan and Alan that did the bulk of the heavy lifting in all of this. It's always been a collaborative effort, and there are in fact more people that we could thank, but these are the critical people, the key people in this project. And then I just want to thank some of the other folks at the Human Genome Sequencing Center, where I work, and the funding for our work. And with that, I will finish. Thank you.